Okay, I'm Talia.
And I'm Alexis, and today we'll be talking about inheritance.
Now, we're going to start with meiosis. Meiosis starts with interphase, and interphase is when the chromosomes are replicated and the chromatin is condensed. That leads to prophase 1, and in prophase 1 we have chromosomes being paired, which is later called homologous chromosomes. Those are being crossed over, giving all of the chromosomes and all of us genetic variation. Metaphase 1 is when the spindle fibers are being attached to the centromere of the chromosome, and those the spindle fibers control the chromosomes and tell it kind of where they go. That is when the spindle fibers line up the chromosomes in the middle or the equator of the cell. And in anaphase 1, the spindle fibers pull back the chromosomes, leading them to the poles of the cell. In telophase 1, the cell is dividing, and the spindle fibers are beginning to form in both of the cells. In prophase 2, we have the chromosomes being condensed. Metaphase 2, we have the chromosomes being lined up in the middle of the cell as the equator. And anaphase 2, we have the spindle fibers pulling back the sister chromatids of each chromosome. And anaphase 2 differs from anaphase 1 because anaphase 2, you pull back the sister chromatids, and in anaphase 1, you pull back the entire chromosome. In telophase 2, you have almost the end product where the cells are being split, and for our final product in cytokinesis is where the cell has now a haploid number of chromosomes in each of the 4 nuclei.
Next, I'm going to be talking about an Austrian monk who is named Gregor Mendel. I'm first talking about his experiment, and with his experiment he used pea plants, male gamete and female gametes, to hand cross-pollinate to learn more about inheritance. One law that he did pronounce from his experiment is the law of segregation, which states that 2 alleles for each trait separate during meiosis. The second law is the law of independent assortment, which states that a random distribution of alleles occurs during gamete formation. Sorry.
Okay, now we're going to talk about Punnett squares. This is a simple Punnett square known as a monohybrid cross. We're going to use a homozygous big B, known as dominant, and we're going to also use a little p, known as recessive. When you do a monohybrid, you use the dominants go on the top, and you use separate columns, and then you use the little p's on the side, known as recessive, as I mentioned before. What you do is you bring down the dominant down first, and which in this case is B, so you do big B for dominant, as well as in this column. And then you bring the recessive across, and so you use a little p here, a little p here, a little p here, and a little p here. And this is pretty much how you would solve a monohybrid Punnett square.
Next, I will be explaining a dihybrid cross Punnett square, which in this case, we're going to say that the parent genotypes are big B, little b, big P, little p, and I'm going to use both traits heterozygous again and be big B, big P, little p. When you determine which 2 traits are picked to go on the top line or the side line of the Punnett square, you just remember this ratio: 1 to 3, 1 to 4, 2 to 3, and 2 to 4. Sorry, that's kind of crammed in here. But what I mean by that is, so let's say this genotype is going to go on the top line. You want to take the first one to the third one, which gives you the first gene. And then you take the first one to the fourth one, which gives you the second one, which is big P, little p. And then it goes on. So the second one to the third one, little b, big P. And then the second one to the fourth one, little b, little p. And you do the same for the top line, which in this case, since they're both heterozygous, both traits, it'd just be the same. Big B, big P, or big B, little b, big P, and little b, little p. This is the part where it kind of gets complicated, but it's really easy if you just follow along. So, like Talia mentioned, you want to stay with the dominant traits first because they go in front of the recessive traits. And what makes a trait dominant is that it shows over the recessive trait, and the recessive trait hides behind the dominant trait. So, you carry down the big B, and then you look to the other side, and that also has a big B. So you put that there, and then those are both big P's, so they both just come down. And the next one would be big B, big B, big P, little p, and then big B, and then this has a recessive B, so it'd come next, and then you have two big B's. And then there's a little p. And this continues on until you fill your square. Just gonna do this real quick.
Okay, awesome. While she's filling out the dihybrid, some key vocabulary words you guys might need to know are allele, as she mentioned. Allele is pretty much an alternative form of a single gene, excuse me, that it may have for a particular trait. And you might need to know what a gene is, and that's pretty much a functional unit that controls inherited trait expressions that is passed on from generation to generation through your family. And you also might need to know what a trait is. A trait is a distinguishing quality or characteristic. And she's going to pretty much finish off this dihybrid cross, and you'll see, she'll later explain what all just happened. Because it does, as she mentioned, it does get confusing.
Okay, I'm gonna pull out an eraser. Erase this part. Okay, so now I'm going to explain, as Talia said, what just happened. So with the two parent genotypes, these are all the possibilities of an outcome of an offspring. You use ratios to show the genotype and phenotype, and the genotype, well, the possibilities of a genotype are big B, big B, big P, big P, big B, big B, big P, little p, big B, little b, big P, little p, big B, little p, little p, little p. I feel like I'm, no, no,
No. You need to do,
oh yeah.
or no. Sorry, I have a little malfunction.
Little b, big P, little b, little p, little p, little p, big P, little p, and so on and so forth. Well, those would be all the different genotypes, and you come through here and you pretty much just like count how many of each there is in this particular Punnett square, and then you write a ratio. Like, this isn't the exact, but for example, it'd be like 0, 3, 1, and so on and so forth. The easier part of solving a Punnett square is finding the phenotype. The phenotype is your outside appearance. It's what
What traits show?
what traits actually show. And this will also take a minute, but the possibilities are, well, let's say that we're talking about bunnies, okay? And the big B is dominant for black fur, which is, the little p is recessive as white fur. So you could have, sorry. Stop collaborating. So you could have a black bunny, and then let's say that the P's stand for ears of length, and big P stands for dominant over short ear, which is the little p. So we're going to say long, and then you can also have black short, and then the recessive, which is white long and white short. And then after that, what you do is you count how many there are. So this is both the dominant traits. They're all dominant traits, so that would be black and long. This would also be black and long. Black and long, 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 black and long. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. So then after you cross those off, you count them up, or you count them up as you cross them off, and that would be the first number to your ratio. And the next would be black and short. So dominant and short, black and short, black and short, and then there's 3. The next one would be white and long. So white and long, white and long, white and long, and then that's 3. And then I'm gonna put the next part over here since it gets cut off, and that's just 1 for white and short. So then your ratio of your outcome of offspring,
which is the phenotype,
is 3, 9, 3, 3, 1. Now, I think it's time to sum it all up.
Yep, it is time to sum everything up.
The struggle. This might take a moment.
Sorry about the wait. I'll go to the computer. I'll click it. I go a lot smoother and faster. Okay, ready?
Yeah.
Story about Mendel, his name is Greg. All day he repeats until he noticed a pattern in the pods and leaves. Inheritance did start by segregation, mononuclear during gamete production, per sperm and per egg, described by Greg. I got described homologous chromosomes, DNA describes genotype, but the outside shows my phenotype, and my fingers touch 101. If I can roll my tongue, try it everyone. So I got a big B with a little b, too many heterozygotes. Dominant on the top line, recessive on the sideline. So I got a big B with 2 little b's, genetic traits we're gonna share. Yeah, 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 it's a party in the Punnett Square. Yeah, 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 it's a party in the Punnett Square. 9, 3, 3, 1 with 2 traits, 16 a hybrid cross. Black or brown, green or yellow, gotta do the test cross. Law of independent assortment, Mendel's hypothesis supported. Try with some pennies, same low probability. Gotta describe homologous chromosomes, DNA describes genotype, but the outside shows my phenotype. Red hair and freckles, or attached earlobe. Do I have dimples? Sure do. I got a big B with a little b, too many heterozygotes. Dominant on the top line, recessive on the sideline. So I got a big B with 2 little p's, genetic traits we're gonna share. Yeah, 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 yeah. It's a party in the Punnett Square. Yeah, 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 yeah. It's a party in the Punnett Square. Feel like making a pedigree, pedigree. Still traits to my family, family. Check out all the disorders, disorders. We look fine, but we might be carriers. So I got a big B with a little B. Too many heterozygotes. Dominant on the top line, recessive on the sideline. So I got a big B with two little B's. Genetics are gonna share, okay. Yeah, 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 yeah. It's a party in the Punnett Square. Yeah, 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 yeah. It's a party in the Punnett Square. So I got a big B with a little B. Too many heterozygotes. Dominant on the top line, recessive on the sideline. So I got a big B with two little B's. Genetic traits are gonna share. Okay. Yeah, 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 yeah. It's a party in the Punnett Square. Yeah, 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 yeah. It's a party in the Punnett Square. Yeah.
Thanks for
Thanks for
watching.
watching. Bye.